Hello, my name is Ashley Cummings, and I'm going to talk about the lac operon. The lac operon is used in E. coli and other bacteria to digest lactose for energy when glucose is not available. The lac operon is fascinating because it uses an operon, which is where multiple genes are controlled under one promoter. Before we get into the model, I wanted to share names of two important proteins in the model. First is calibrate activator protein, or I will refer to it as CAP. And what CAP does is it helps RNA polymerase bind to the promoter sequence of DNA to begin starting transcription when lactose levels are high. Our other enzyme is the LAC repressor protein, or LAC, and LAC inhibits RNA polymerase from transcribing by binding to the operator sequence of the DNA and thus inhibits it from doing transcription. And it does this when the glucose levels are high. Now, a few disclaimers before I get into the model. I will not be talking about the proteins that the lac operon transcribes for because of time's sake, and I will also be only showing 2 out of the 3 genes in the model just to make the model a little bit more simpler. And then, as we can see here, RNA polymerase is in the shape of an arrow, and that is to signify the direction that transcription would be occurring. So here we have our inactive model of the lac operon. And we see here that these boxes are what represent our DNA sequences. And then we have CAP here, LAC here, RNA polymerase, and the small molecules, the small purple shapes are called glucose. And what makes this model inactive is that glucose is present. If glucose is present, E. coli will always break down that first because it's a smaller molecule to digest. And because of the presence of glucose, this means LAC is active. And when it's active, it binds to the operator sequence shown here. And it blocks RNA polymerase from binding to the promoter sequence, and thus it cannot do transcription of the lac operon. Seen here, we have the model of the lac operon when it is active. And this is seen by the larger molecule here. Lactose is present. And when lactose is present, a rearranged version of lactose called allolactase binds to lac and changes the shape. And this shape is what inhibits it from binding to the operator. And because the glucose levels are lower or non-existent, CAP can bind to the CAP binding site, help RNA polymerase bind to the promoter, and RNA polymerase can begin transcribing the genes needed to digest and transport lactose. So the lac operon is overall an amazing example of how bacteria like E. coli can transcribe certain genes to help them survive in their environment depending on the levels of glucose and lactose present. This is also exciting because of modifications that can be made to bacterial DNA. For example, genetically engineering bacteria to make insulin to help diabetic patients with type 1 diabetes. Here are my references, and thank you all for listening.